Puppies.
Nope.
HIV.
We don't talk about dogs in here unless it's about vaccines and autism, which apparently people are arguing in dogs now too.
That's just dumb.
Yeah. Thank you for allowing me to, or depression in dogs. Thank you for allowing me to kill the mood. All right. Oh, where are we at? Okay, so couple of things for Friday. If you want to drop your poster off with me before Friday, do so. There's a couple of groups that I still don't have a final copy from electronically, so send those to me. 9 o'clock downstairs, be ready to go. Attire, I don't really care. This is perfectly fine what you're wearing now. It's your choice. I always say let the research do the talking instead of what you're wearing. So if you're doing a shirt and tie, you're trying too hard.
I say we all wear lab coats. That's mine.
So that's the first thing. Second thing is, I know I've forgotten to mention this the past couple of times, but if you remember the extra credit opportunity, 3 questions related to Ebola or HIV through what we've covered at the end of today. And those are potential questions for the exam. In a perfect world, what I'd like to have is 1 simple, 1 medium, and 1 hard question written by you. And these should be multiple choice questions. And I'm not going to necessarily, if you can put down 4 options, that's great in identifying the right one. If you can just give me the right answer, that's fine. I'll do the rest of the work. But anything to just kind of give me a leg up for next week would be awesome. No more assignments. We're not going to do a lit review or a critical thinking next week. So the plan will be on Monday to finish up anything on HIV that we haven't talked about, and then Wednesday and the rest of Monday to be working through additional review questions on HIV. Okay? And then exam next week Friday, and then we'll talk about that last week sometime next week. I think that's all I have. Are there any questions right now? Grace.
Is exam 3 the same setup as the other ones?
Mm-hmm.
Okay.
Exact same. So I will actually be starting to work on the take-home portion probably today or tomorrow. More likely today or Friday. Tomorrow's a day off because it's Jen's birthday, and I shouldn't work on Jen's birthday.
Aww.
So plan that I'll be working on that, and probably my hope is I'll be able to get that to you guys by Wednesday, so that you have 9 days or so to work on it. Yeah. Other questions? Okay, so where we left off on, where we ended up leaving off on Monday was talking about the structure here of the DNA version of the genome once it's integrated, and this is just a map of it again. What this image is showing you is roughly the gene coding regions. And again, you may remember that the 5' end has an LTR which contains U3, R, and U5. The 3' end includes that as well. And then the one thing that I wanted to point out here real quick is, this is actually, now that I'm looking at this, I think that this is showing in part the transcript as well. But here's the promoter region, and we said that host genes are controlling this promotion of transcription. So these are going to be the initiators of transcription. And then we said that this TAP protein, I'm going to kick this today probably, this TAP protein from HIV binds to that transactivating region, which is the stem loop structure. What is the function of TAP? What does it allow for? What's that? And what does that phosphorylation do, Sabrina?
Keeps the RNA polymerase going and initiating.
It allows for the elongation process. Absolutely. So TAP binds to TEP-B, which includes these proteins, cyclin T1 and CDK9. Those phosphorylate the RNA polymerase and allow for this elongation. There is another element here that's pretty important that we haven't talked about, and that's this what's known as the RRE, or Rev responsive element. This will be found in, this is actually an RNA sequence that is bound by the viral gene, or the viral protein Rev. I'll give a picture a little bit later to explain exactly how this is working. But this is important for ensuring that full-length, unspliced, or incompletely spliced mRNAs will be able to make their way outside of the nucleus. So this is how the virus ensures that full-length genomes will exit in order for those to either become the genomic RNA or for those to be translated into GAG and GAG and PAL. You'll notice a couple of features here just looking at this. So the first thing is the GAG and the PAL transcripts, or the open reading frames, actually overlap with each other. This is where the ribosome stuttering is going to occur to allow for either a GAG or a GAG-PAL polyprotein to be produced. Rev, TAT, are two where we see alternative splicing occurring. And so these two will be different mRNA products that are a result of mRNA splicing. And we talked about a couple of other mechanisms that are used here to ensure or to regulate which genes are being expressed and how much of each of them is being expressed. So other examples, there's thought to be potentially an iris, which allows for cap-independent translation. And if that iris exists, it exists somewhere within this U5 or right around that TAR transactivating region. There are thought to be, what were the other ones that we listed off? Polyprotein, potential iris, ribosome, slippage, alternative splicing, and then ribosome shunting, which is the last one that's thought to be involved, but we're not going to go back and recover that. Here's just giving you an idea of all the potential transcripts that have been mapped. Okay, by no means do you need to memorize these, but all of these down here are ones where we have subgenomic RNAs, so these would be mRNAs that are processed by alternative splicing. Note the one thing in common is that they all contain this beginning of the sequence right here, from where the cap is, and then a little bit into the U5 region. Those that undergo alternative splicing lack the GAG and the PAL proteins. And so this includes transcription of the accessory proteins like VIF, VPR, TAT, REV, etc., or include the envelope, the GP120 and the GP41. I'm going to pull up a picture real quick with hopes to explain how does this regulation occur. Alright, so here's what's thought to occur. When levels of, this seems to in part be controlled by how much REV is expressed within the cell. So when levels of REV are really low, what we find is an abundance of the spliced forms of the genes, or the alternatively spliced forms. So what this image here is showing you is that we have three potential types of RNA species that will be produced. Ones that are unspliced, the full-length genomic RNA. Ones that are partially spliced, and notice both of these contain that Rev responsive element. So these partially spliced ones would actually include the envelope mRNAs, mRNAs that contain the envelope gene. And then we have ones that are fully spliced, that lack this RRE. These ones, in the absence of Rev, these smallest ones are the ones that actually are able to exit the nucleus because they look like a host mRNA. They've undergone splicing, they've undergone 5' kappa addition, poly A tail addition, and so they're ready to be exported. The genes that follow this process, if we go back to that original, or to the PowerPoint real quick, the genes that would then be exiting the nucleus in this mRNA fully spliced form would include TAT, REV, and NEF. Would not include those that contain a REV responsive element like VIF, VPR, and the envelope. Okay. So what this allows for then is that in low expression, this is of REV, this is selectively allowing for 3 genes to be expressed. And you can kind of consider these to be analogous to like early genes that we talked about with bacteriophages. These are ones that are going to be essential for the initiation of the lytic cycle again. So for example, TAT would be involved in increasing gene expression or allowing for elongation of transcription. REV would be involved in allowing for the envelope or the GAG and the POL genes to be transcribed. When levels of REV increase then, what we find is a bit of a shift. As levels of REV increase, REV is able to bind to this RNA, to this REV responsive element, and now allow or help to ferry these 2 larger size species outside of the nucleus through nuclear pores. Okay, so REV seems to be essential for the export of these 2 larger products. Once they exit, REV will actually detach, re-enter the nucleus, and continue to shuttle or ferry more mRNAs across. So REV is kind of catalytic in this way. It will continue to work. It's not as if it exits the nucleus and then gets degraded. It will re-enter, run the process all over again. Jonah.
So is it grabbing like unspliced mRNA or spliced? Or?
It's grabbing it's grabbing RNAs that have the REV responsive element. So those would be unspliced, or those would be partially spliced RNAs. So again, going back to our original picture here, those that contain VIF, VPR, or the envelope, these would all be considered our partially spliced products. Those all contain a REV responsive element, as does the full length RNA as well. Okay. Sabrina.
So is that just because without REV, the unspliced and partially spliced would not leave it?
Exactly. That's my understanding. Now, what exactly prevents it? What exactly distinguishes a partially spliced from a spliced? I don't really know the answer to that. I don't know if there's something about the REV responsive element that just blocks those ones from being exported. I couldn't give you a clear answer because I can't find literature to say why. The full-length one, the argument there is that that eukaryotic mRNAs have a mechanism to prevent unspliced RNA from exiting. So, and again, I'm not exactly sure how it gets identified. My guess is it has to do something with the sequences that usually are at the 5' and 3' ends of splice locations. So maybe it has something to do with recognizing those splice sequences still being embedded in the RNA. I mean, you could view this as being a positive in eukaryotic mRNAs. All of ours are undergoing splicing before they're exited from the nucleus. If not, there's not going to be a functional protein made, right? So it suggests that there's some mechanism in there to ensure splicing has been completed. But what controls that? I'd have to take a look at the literature. I don't really know. Grace.
Why does it leave and then like come back in, I guess?
So the story's a little bit more complex than just it leaves and comes back in, but it interacts with a protein known as RAN-GTP, and RAN-GTP is the actual protein that will shuttle the whole thing across. Rev also has what we call a nuclear localization signal, which allows it to come back in. So Rev interacts with RAN-GTP when RAN-GTP exports through one of the exportant channels. It actually will be dephosphorylated, so it goes from a GTP to a GDP form. That allows it to re-enter into the nucleus, and Rev is re-entering along with it. So it seems that what Rev is really doing is interacting with RAN, and as RAN is moving across, Rev is being captured along with it.
So it's not the genomic material that's like getting put back in? Is it just Rev?
It's just Rev.
Oh,
Yes,
that's what I was
it's just
confused
Rev.
on.
No, so once you have exit, Rev seems to detach or disassociate from the RNA and then re-enters on its own.
Okay.
Yes,
thought, like,
sorry about that.
yeah,
No.
you're fine.
Okay. Yeah, so it's just Rev that's coming back in. I want to point out here too, one of the features that we see in the full-length RNA forms, and that is that notice all of these here, these blue arrows, these are all cleavage sites. So the GAG and the GAG-PAL precursors are actually produced as polyproteins. HIV's got a protease that will cleave these and resolve them. And actually, some of that cleavage doesn't really occur until during the budding or just after the exit. So there's one single mRNA that produces both the GAG and the GAG-PAL polyproteins. And so what that indicates to us then that the mechanism for producing GAG or GAG-PAL has to come from something in the translation process. And that is ribosome stuttering. We're not going to go into this in great detail because the mechanism can be a little bit tricky to understand. I just want to kind of point out a couple of features here. Ribosome stuttering or slippage is an example where the ribosome will basically, like we slip on ice, will kind of slide forward or slide backward along an mRNA sequence. Particularly, there's two things that have to be in place for this to occur. The first is what we call the slippery sequence. It is a run of U's. And when we have multiple nucleotides that are identical in a row, oftentimes that leads to slippage not only of the ribosome, but we see this being an issue for RNA and DNA polymerases as well. Actually, these regions where we have maybe a run of A's or a run of T's are areas where we most often find insertions and deletions occurring in the genome. Okay? Because the polymerase kind of loses track of where it's at. So in this case, what we have are overlapping reading frames between GAG and PAL. GAG would read a phenylalanine leucine by the ribosome and would just continue to read these in 3 nucleotide steps. The other feature that's required is a stem loop structure. And basically what the ribosome does is it bumps into this and gets knocked out of its reading frame. In other words, the ribosome, you can imagine, is quite large, right? So we've got, here's our ribosome. Here's where we might expect to see the A site, where a tRNA would be coming in. And the ribosome basically, like a bird into my car this morning, runs right into the hairpin loop structure. Oh, and it wasn't one bird, it was 2. Sorry.
Are you moving?
Yes, yes.
Is your windshield shattered?
No, they hit the body of the car. So instead of killing 2 birds with 1 stone, kill 2 birds with 1 car.
Murder.
I know, they were fighting though. So,
Were they fighting?
I think so. Highlight of my morning. All right. So instead of reading phenylalanine leucine, what happens is that stem loop structure, basically, the ribosome kind of bounces off of it, backtracks a nucleotide, and now it switches the reading frame. So instead of continuing to read GAG and then stopping at a stop codon further down, what happens is it changes the reading frame and now allows for GAG and POL to be produced as a single polypeptide. This happens maybe only 1 out of every 20 times. Frequently, GAG is produced. 5% of the time, GAG-POL is produced. Sabrina. Yeah, I can try. Okay. So again, what happens is the ribosome is reading this slippery or coming up to this slippery sequence. It might run into this stem loop structure, and instead of kind of holding on to the slippery sequence correctly, kind of bounces backwards 1 nucleotide. Switching the reading frame from it, instead of reading a UUU, UUA, it would read UUU, UUU, AGG, so on and so forth. Okay. The mechanism for how this occurs is not fully worked out, but it does seem that the slippery sequence and the stem loop are required. So GAG is produced more often. In other words, 95% of the time, the ribosome doesn't slip. The slippage is a rare event. Okay, so that slipping, it happens about 5% of the time to produce the GAG-POL polyprotein. Yep, so that's produced 5% of the time. Majority of the time, it's just going to be GAG. Right.
GAG comes first?
Yes, because GAG comes first in terms of the sequence. And ultimately, in that case, then what we're going to find is we're going to get stopping of translation if we continue to read the GAG sequence, phenylalanine, leucine, whatever GGG is, AAG, AUC, so on and so forth. Most of the time, the ribosome will resolve this hairpin loop and help to break these bonds, and it will continue reading until it hits a stop codon and stop translating GAG at that point. But again, on some rare case, we get this frame shift occurring. That means that the 3' or the end of the GAG sequence and those GAG-POL polyproteins is actually slightly different than when it's just GAG itself. Right? Jonah.
If you move the ribosome by like 1 nucleotide, you shift the reading frame, why not mix up the protein?
That's what I'm trying to. Yeah, that it could mess up the protein. So, but it wouldn't mess up all of the protein. It wouldn't mess up all of it. It would only mess up the part where that sequence is different. So look at what actually HIV has. If we kind of take a look at this region right here. There's a little bit of a spacer sequence of polypeptide that does nothing. So the reason the region where this ribosome slippage occurs is actually really encoding for just what we might call a linker polypeptide. A region that doesn't actually have any particular function other than to link GAG and POL together. So yeah, it changes the amino acid sequence, but does it disrupt the proteins? No. It's kind of a buffer to prevent any mutation or change in the amino acid structure or protein structure to those GAG proteins or to those polymerase proteins. Grace.
So, because the slippage only happens 5% of the time,
Yeah.
is that why HIV, is that dormant in the period of its infection?
Not necessarily. What this does is it allows for difference in the amount of those GAG proteins and the polymerase proteins that are going to be produced. That dormancy is more a product of the NF-kappa B and the SP1 proteins being expressed from the host. So think about this in terms of the virus real quick, though. What do the GAG proteins encode for? So there's an MA, there's a CA, and there's an NC. What are those? CA could be a giveaway. That's the start of a word. Go with it. Oh, good Lord, thank you.
Oh.
Capsid.
Cat.
Oh, you said cat? Well, that's totally not correct. Capsid. NC stands for nucleocapsid.
Oh.
MA stands for matrix. Okay? These are all capsid proteins. And what are oftentimes the most abundant proteins in a viral particle? It's the capsid proteins. So it should make some sense that this is the order in which these two are made. GAG is produced the majority of the time, and then 5% of the time when GAG is produced, so are the polymerase genes, or the Pol genes, which include the reverse transcriptase, the integrase, and the protease. You really don't need many of these, because these are enzymes, and enzymes function catalytically, and we only need a couple of them in each viral particle. Okay? So what this is saying is, if you're thinking about stoichiometry real quick, you've basically got about 20 GAG proteins, each of the nucleocapsid, the capsid, and the matrix protein, for every one reverse transcriptase that's produced. Stoichiometrically, that's what's happening here. And that should make some sense, that we would need more of those capsid proteins.
What does PR stand for?
PR is protease. So that's the actual enzyme that will perform the cutting of the matrix and the capsid and the nucleocapsid and the RT and all of these other ones. Okay. Sabrina, do you have a question?
So, is the reason that the protease is packaged into virion because that maturation doesn't occur until it's...
Yeah, and you could, yes, and you could make some arguments about a conservation or the importance of this from an evolutionary standpoint. We'll talk about that with the maturation a little bit later, but think of this in maybe a slightly different way. These are produced as polyproteins. One of the things that the matrix protein does, it actually gets modified. It gets a lipid attached to it, which helps it to embed itself peripherally to the inner face of the cell membrane. Well, since it's a polyprotein, what comes with it? Huh? Protease and the reverse transcriptase and integrase and capsid and nucleocapsid. All of those come with it. So it actually, you could see where this would be advantageous in that it makes the recruitment of all those proteins to the site where budding is going to occur, makes that really, really easy to do.
Self-assembly?
Yeah, it really helps out with the self-assembly. And speaking of which, so here we are. I'm going to see if there's anything else I wanted to cover there that I've missed. No. Okay. So I'm going to kind of go off script here a little bit. After we talk about this assembly and maturation, I want to talk about a couple of specific features. But basically, the virus, or at least one of the models right now, is that the viral particle exits still being immature, and it needs to undergo a final maturation process in order to become infectious again. And that's the role of protease. So the protease enzyme, which they're showing in this here, it's this light blue image. Oh, sorry, that I just circled virion. Protease, this light blue one right here. And you can see we've got the matrix and the capsid and the nucleocapsid proteins all right attached to each other. We've got at some lower frequency the protease, the integrase, which is shown in blue, and then that gray blob is the reverse transcriptase. And everything is still attached to each other. Protease now begins to cut these into their individual pieces to allow for or finalize the self-assembly of the capsid proteins into that icosahedral-like shape, or to allow for the self-assembly of the nucleocapsids on the RNA genomes. What could be... The question of why in biology is always tough to answer, but we can at least think about why might this mechanism exist. So why might HIV have this delayed maturation as a process? We haven't seen this with other viruses. Jonah?
So will they infect the same cell?
That's the argument that most individuals make, is that it prevents the reinfection of the same cell. And that cell usually, if the virus is undergoing lytic replication, is going to die. So it just prevents the reinfection of a cell that's already on its way out. Now, a couple of things to point out here about this exit. Actually, I'm going to pause there real quick and just see if there are any questions. Go ahead, Jonah.
I was going to ask if GP120 and 41 bind to the infected cell, the third proteins are not expressed, or like the proteins that...
CD4 is repressed. Yeah, so we might argue that part of the reason why there isn't a reattachment also is because CD4 is repressed. The other thing is GP120 and GP41 actually have to undergo a proteolytic cleavage as well. They don't undergo that cleavage until during the exit process. Yeah, that's a good question. Now, a host protein actually makes a reappearance here, one that we talked about with Ebola. Anybody remember the name of the protein that helps to prevent Ebola from budding? And VP35 has a mechanism of repressing it. We talked about, thank you. What was that? Tetherball 1. Tetherin. Exactly. I was going to bring up tetherball, so I'm glad you did. So I'm just going to kind of go back a number of slides real quick, back to one of our initial images of this process. And this is a little small, so I apologize, but the middle of this image here is showing a couple of things that's happening. It's showing that NEF and VPU are both inhibiting the expression of CD4. The other thing that it's showing is that VPU is actually inhibiting the export of tetherin to the cell surface. And so here we have another example of a virus that inhibits the expression of tetherin. In Ebola, it was VP35. In the case of HIV, it's the VPU protein. You could make an argument that maybe the two of them can functionally... is it VP35? I feel like I'm saying that incorrectly. I got to double check that on Ebola. But you could make an argument that the Ebola protein that blocks tetherin expression and VPU might be able to actually substitute for one another because of this common function. It's either 35 or 24 or 40. I can't remember. It might be 40.
35, I think.
We'll figure that out. We'll come back to that. I want to make sure we have that right. Sounds like a good exam question. Not everyone should write it. All right. Okay, what I'd like to do is just kind of point out a couple of things about HIV with this process of exit that we haven't really talked about, and this kind of actually brings us back to some questions that were asked. Oops. Oh, okay. Asked at the beginning of this, which was, I know some people had asked questions like, well, why is GAG and POL being produced from the full-length genomic RNA? And how does the virus decide whether that full-length genomic RNA is being used for translation or being packaged as a genome? And the answer to that second question, how does the virus decide, it seems to be random, is the simple answer. Nobody has come up with the rationale for HIV, how it gets determined whether a full-length genomic RNA is going to be used for translation or to be packaged in viral particles. Actually, there is evidence to suggest that a full-length RNA can serve both functions, be translated and then be packaged later on. Okay. The packaging is pretty interesting here, and what I want to do is I want to go back to another picture that I found in an article that I think could be helpful. So here's just looking at the genomic RNA again. This is a different version of it, but you'll notice here's the remainder of an LTR, the R and U5 region, U3 and R poly A tail on the end. And this right here is thought to be in part the iris structure. Okay? We have our TAR. We have the binding site for tRNA lysine, which is also going to be incorporated into those capsids. We've also got a couple of other features. What's known as a Psi or a PSI site. This is thought to be involved in packaging. And then we have a dimerization domain. What do we know about how many genomes of HIV are packaged into every virion? 2. So what's the role of this dimerization domain? Allows for those 2 genomes to actually bind together. Note that this is a stem loop structure. That means that there's homology on both of these ends. What it seems is that when this secondary structure gets disrupted, what it allows for is those 2 mRNAs to actually attach to each other at the 5' end. So we actually get base pairing between those 2 genomic RNAs, and that ensures that 2 are going to be packaged in every viral particle. Okay. This Psi region here, to make a long story short, this also contributes to the recruitment of the genomic DNA into the genomes. And what is thought to occur is that this region is actually bound by the nucleocapsid portion of that GAG polyprotein. So GAG, that nucleocapsid region, would bind to this packaging site first, and this allows then for nucleocapsid to begin polymerizing or multimerizing on the surface of those RNAs. So we get multiple nucleocapsids located. We've also got our capsids attached to each of these. And finally, those matrix proteins. What do the matrix proteins have to interact with? They're going to be embedded in the membrane, partially. What else is in the membrane? Not CD4, but what that interacts with CD4. GP120 and GP41 will also be in the membrane. Matrix protein has 2 interactions. One is with the capsid protein, and the other, it interacts with this cytoplasmic region of the envelope proteins, GP120 and GP41. That interaction allows for then the matrix to recruit everything to these regions where that envelope protein is found. Okay, so that's allowing for that self-assembly to occur. I think that's all I've got. Questions?
What does the matrix protein do?
So the matrix protein, again, is part of that GAG protein, so the one that makes up the capsid, etc. The matrix protein is there to recruit the capsid and the nucleocapsid and the RNA polymerase to sites where we've got GP120 and GP41. So matrix protein is essentially a scaffold protein to help that self-assembly process.
So are GP120 already in the membrane?
They would be already in the membrane, yeah. By this point where we are getting this self-assembly occurring, Sabrina, GP120 and GP41 would be embedded in the membrane by this point. Shania.
So the dimerization of the iris, you said that ensures that two genomes are per virion?
Yeah. So the dimerization has palindromic sequence. So for example, I'll write out let's do a whiteboard. Apparently something fun is going on in the hallway. So here would be an example of a hairpin loop sequence where the A and the U could base pair, the U and the A, the U and the A, and the CC would be the stem of that stem loop structure. Well, if you have another, and let's just say then this is like the 3' end over here. If you have another mRNA, with the same sequence at that dimerization region, you can get at least partial base pairing. Okay, so this would be an example where that dimerization domain, because of that stem loop structure, allows, instead of intramolecular binding or formation of a stem loop in a genomic RNA, single molecule genomic RNA, it allows for intermolecular binding to occur, or base pairing to occur. That dimerization then ensures that those two genomes would be packaged together. Does that help, Shania?
Yeah.
Okay. Other questions? Jonah?
How does the dimerization hairpin thing classify as an iris? I thought that was differentiated in transport.
Translation? Yeah, it is. But nothing says that an iris can't be more than one thing. Right? An iris is kind of ambiguously defined. We don't really, there's no common structure to an iris. We identify them by testing them. The most common feature is that it's a secondary pattern or secondary folding of RNA back on itself. But if you look at the iris of HIV versus the iris of something like hepatitis C, or even poliovirus, they look completely different from one another. There's nothing that's common except for the fact that there's this highly intricate 3-dimensional and 2-dimensional secondary structure of the RNA. So what this is saying is that in this case here, at least, we're finding that there is that this region is serving multiple functions. Not only is it functioning as an iris, but it also is functioning as a packaging signal. And it's also functioning as a dimerization signal. And it also contains the TAR as well. Now, I think whether this is a true iris or not is still up for debate. I could see where there would be where there might be an advantage to having it, but at the same time, the mRNA looks like the host mRNA to begin with. It's got a 5' cap. It's transcribed by host RNA polymerase, and so you could argue, what would there be a need for an iris, or why would there be a need for an iris? Maybe it's important for the late stages of replication when we start to get dimerization of those RNA genomes. I don't know the answer. And you could at least make hypotheses why it might encode them, though. Okay. Sabrina.
So the intermolecular base pairing that you drew, that's between two full-length RNAs?
Yes, that would be between two full-length RNAs. So it would be as if you've got this, so let's say that this one drawn in this image here is one of those. You would have another one with its cap here. Here's where you're getting some of that intermolecular base pairing, and then the remainder of its sequence with a poly A tail. Any other questions? So I think what I'll leave today off with is then a couple of things. One, let's go back to the big picture and think about the life cycle just briefly. So taking a look at the life cycle again, the attachment is between GP120 and CD4 and that co-receptor. The one we've primarily talked about is CCR5. We get entry by fusion. Reverse transcription is occurring in partially uncoated capsids. As this is moving along microtubules to the nucleus. And what happens in the nucleus? What's one of the first steps that takes place? What has to happen to the viral genome? What's that?
Reverse transcription.
That'd be occurring here in the cytoplasm. Integration with the enzyme integrase. And then we have this latency period, which we kind of just glossed over for right now, because really nothing's happening. But as the host replicates, the viral genome replicates along. And then we start to get transcription, which is what we talked about today. And transcription first is going to occur, or we're going to see first biosynthesis. So transcription and translation of proteins like NEF, VPR, and the key one, or I'm sorry, NEF, TAT, and REV. And TAT and REV are kind of the major ones here. TAT's going to help to enhance elongation of transcription. REV is going to help to export those full-length or partially spliced mRNAs. And that's where we start to get then the production of those structural proteins, the polymerase, the reverse transcriptase, the integrase, the protease, the GAG proteins, and the envelope proteins. The assembly is going to occur on the membrane. We get a budding process, and then maturation is thought to occur sometime after the budding has been completed. And again, that maturation requires the protease. Now, the reason why I take a minute or two to walk through this is, one, just to kind of put this into the context of a full infection. But secondly, all of these steps are actually targets for retroviral drugs, and that's where we're going with this next. So, for example, fusion can be blocked by drugs like Cell's Entry, which block the interaction between CCR5 and GP120. Or the integration can be blocked with integrase inhibitors. Reverse transcription we have more drugs for reverse transcription inhibition than any other of these groups. These were kind of the easiest to identify because a lot of them were already sitting on the shelves. We also have drugs that can block the process of maturation, protease inhibitors. Okay. So there's multiple points at which antiretroviral drugs can work. And I think we've alluded to this a little bit already, but treatment these days is not just with any one drug, it's with a combination of 2 or 3 different inhibitors at different select points of the pathway. Okay. So on on Monday, what I would like to do is I've got a couple of slides that I just want to use to talk about antiretroviral therapy and the evolution of HIV, which can lead to resistance. And then we're just going to spend the rest of the time looking at some specific examples and talking about what is the effect of these drugs on the life cycle? All right, have a great day. If you have any questions about Friday between now and then, let me know, okay? I'll email it if it's important to everyone. Key thing, have fun. All right, have a good rest of the day.